Good afternoon, everyone. My name is Irene Cobo. I'm a postdoc at the Department of Ecology and Evolutionary Biology at the University of Connecticut. And I'm here today to talk about CartograPlant, which is a cyberinfrastructure which objective is to help improve plant health and productivity in the context of a changing climate. So CartograPlant is basically a web-based application that integrates, visualizes, and analyzes genotypic, phenotypic, and environmental data from georeferenced plants, including forestries. And this application is relevant and timely nowadays because, as we all know, climate change is threatening plant health and productivity at all spatial scales. However, it remains largely unknown whether plant breeding can keep pace with the rate and direction of this environmental change. On the other hand, the incidence of invasive pests and pathogens is increasing due to globalization, and this tendency is being exacerbated by this climate change. In addition, another threat specific to forestries is the illegal logging and deforestation. And as a result, future plant health and productivity will depend on the match between the genotypes, phenotypes, and the new environments. Because this match will allow the identification of genes controlling traits that provide tolerance to these biotic and abiotic stresses. And these genes can be used to the design of conservation and management strategies. On the other hand, the illegal logging and deforestation can be tackled by identifying the timber origin using chemical, genetic, and anatomic tree data. As a result, tools that collect, integrate, and facilitate this data, such as CartograPlant, are critical nowadays. Regarding the data integration, because as I have mentioned before, CartograPlant integrates, visualizes, and analyzes data. There are three main data types integrated in CartograPlant that are genotypic, phenotypic, and environmental, as I have mentioned before. However, the question is, how can these disparate data types coming from different studies be integrated? The answer is thanks to the metadata collection using ontologies and standards. These data come from two different sources in CartograPlant. One of these sources is the direct submission of studies by users using the Triple Plant Object Submit or Tpps Pipeline. And the other source is the biocuration effort of the affiliated databases. Regarding the Tpps Pipeline, it is basically a pipeline that collects genotypic, phenotypic, and environmental data and the associated metadata from published or soon to be published studies and provides a DOI to the user. It hosts population genomics, association mapping, and landscape genomics studies. And regarding the metadata, it is collected using ontologies and standards, as I have mentioned before, including the minimal information about a plant phenotypic experiment or MIAPI. This metadata collection ensures the so-called fairness of the data, which is the findability, accessibility, interoperability, and reusability. And finally, this pipeline supports standard genotypic file formats such as BCF, which is very convenient for the users. These are examples of studies that are that have been uploaded to CartograPlant thanks to the Tpps pipeline. And it is worth mentioning that the vast majority of these Tpps submitted studies are available in CartograPlant thanks to the efforts of our biocuration team. Regarding the environmental data, it is also available in CartograPlant in the shape of environmental layers. And currently, CartograPlant hosts a total of 914 environmental layers, providing all this environmental information, which is very useful for users in order to ask a wide diversity of biological questions. Regarding the CartograPlant current statistics, CartograPlant currently hosts about 8.3 million of plants, belonging to 500 species, 200 genera, 43 countries, and a total of 179 studies. And as a result, there are available a total of 59 million of genotypes, 1.3 million of phenotypes, and as I have mentioned before, 914 environmental layers. Regarding the data visualization, because as I have mentioned before, CartograPlant integrates, visualizes, and analyzes data. So I have just explained the data integration, and I'm going to explain now the data visualization. The CartograPlant interface is divided into two panels. The left panel, which is the action panel, where users can interact with the plants and the map, and it is located on the left part of the screen. And the right panel, which is a worldwide interactive map that shows the plants and the environmental layers selected on the left panel. So for instance, here, regarding the environmental layer visualization, users can visualize these environmental layers by clicking this button here. And by doing so, all the environmental layers available in CartograPlant are displayed. And here, for instance, we are displaying the major soil groups. And as I have mentioned before, the worldwide map is interactive. So by clicking directly on the map, users can display this white window that shows the latitude, longitude, and the elevation information at this specific point. And also in this case, we are displaying an environmental layer showing major soil groups. Users can display the soil type at this specific point and also a brief description about this soil type. In addition, users can also display information about this specific plant in the worldwide map by clicking directly on it. And again, a little window is displayed here, as you can see, showing the latitude, longitude, and the elevation information. Also a picture of the plant and taxonomical information such as the family and the species. And also you can see two buttons here, one in green and another in yellow. The green button, by clicking on it, users can select this plant for further data analysis. And by clicking on the yellow button, users can display information about the study to which this plant belongs. So as you can see here, users can display the study name, also the authors of the study, the year of publication of the study, and also users can download data belonging to this study, in this case, for instance, phenotypic and genotypic information. Finally, users can also select the plants for further data analysis, and it can be done in two different ways. One way is in the left panel by filtering the plants using different criteria, such as, for instance, taxonomical information, family, genus, species, the markers, plant structure, phenotypic attribute, and also study title, author, and accession. Another way to select plants for further data analysis is by drawing directly a polygon in the map using these buttons here. And once the plants are selected, authenticated users, because in order to perform data analysis, it is necessary to navigate authenticated. Authenticated users can perform data analysis by clicking in this Analyze button. So when users click this button here, this window saying Cartograph Analysis is displayed. And as you can see, there are several tabs there. So the first tab shows the phenotypic or the traits that contains the selected plants. And users can select the traits or the phenotypes of interest for further data analysis here. And in order to help this decision, this tab shows the overlapping phenotypes among the selected plants. In addition, users can also filter the genotypic data that they want to be used for further data analysis. And again, in order to help this decision, we are working in developing interactive visuals and graphs and histograms in order to help this genotypic selection. For instance, we are working in developing a visual like this to help users decide the SNP selection. We are focusing mostly on SNPs because they are the most widely used markers nowadays. But we are also working in providing these visuals for other types of markers, such as microsatellites. So here, for instance, users can select the SNPs based on the to the study to which these SNPs belong, and also based on their overlapping status among studies. Also, we are working on developing histograms, interactive histograms, in order to filter these SNPs and the individuals used for data analysis by quality. And finally, the last tab allows users to select the environmental data of interest for further data analysis. So in this tab, all the environmental layers available in CartograPlant are displayed, and users can select the environmental layers that they want to use for further data analysis. And finally, in the last tab, users can also select the analytical workflows that they want to use for data analysis. This data analysis, well, as you can see, these workflows provide there is a brief description of the workflow, and also these workflows will guide the users, will provide guidelines to the users in order to decide the best analytical thresholds or decisions for their data. But we are still working on it. This data analysis, it is possible thanks to Galaxy, because CartograPlant is connected to a Galaxy instance. And Galaxy is a user-friendly program that allows users to perform bioinformatic analysis without the need of coding skills. Here in this picture, there are represented all the analytical workflows that are available in CartograPlant, or that they will be available soon in CartograPlant. And highlighted in red are the workflows that are fully available, and in black are the workflows that we are still working on. So as I have mentioned before, in CartograPlant, there are phenotypic, genotypic, and environmental data. Regarding the genotypic data, the workflows are mostly focused on SNPs, because again, they are the most popular SNPs, molecular markers nowadays, but we are also working in providing workflows for other marker types such as microsatellites. So the first workflow is the SNP quality filtering that allows users to filter the SNPs by quality in order to ensure the trustworthiness of the results. In addition, we also provide a SNP imputation workflow that allows to infer the lacking SNPs. And this way, we enhance the association power of further analysis. In addition, we are also working in providing a workflow for SNP remapping to allow users to remap the SNPs against the latest version of the reference genome in order to have the SNPs the most updated as possible, and also to have the SNPs mapped against the same version of the reference genome, which is necessary to perform certain analysis, such as meta-analysis, for instance. We also provide a population structure and kinship workflow, which is interesting by itself in order to ask population genomic questions, but also it is necessary to perform association mapping and landscape genomics analysis, because it is necessary to correlate by population structure and kinship in order to avoid the spurious associations. In addition, we also provide association mapping and landscape genomics analysis. So the association mapping is basically the association between the phenotypes of interest and genotypes, and allows users to obtain candidate genes of resilience, for instance, to biotic and abiotic stresses. And landscape genomics, which is the association of the genotypes and environmental variables of interest, and allows users to obtain locally adapted SNPs. And finally, by using the summary statistics of these two analyses, association mapping and landscape genomics, users can also perform meta-analysis. And we are working on providing a meta-analysis workflow. So a meta-analysis is basically an analysis using the results of several studies. And it is interesting for researchers because it allows to increase the sample size and also the number of markers used for the analysis, and as a result, the association power of the analysis. This meta-analysis is possible thanks to the metadata collection in CartograPlant using these ontologies and standards. So as a conclusion, we can conclude based on all this information that CartograPlant is intended to serve as a community resource for plant molecular ecology. And regarding our future directions, we are mostly focused on providing robust and flexible analytical workflows to allow users to ask a wide diversity of biological questions and to analyze a wide diversity of data types. And we are mostly focused in providing a SNP remapping workflow and a meta-analysis workflow. And we are also planning to incorporate a population structure visualization as a loaded layer in the worldwide map, which allows users to put the population structure information into an environmental and geographical context. And finally, we are also planning to do a virtual training workshop for CartograPlant in June 10, 2022, and a context for data submission via Tpps. So please stay up to date to these events in our webs and social media. And finally, I would like to thank all the members of the project that have made this project possible, also the funding institution of this project, and to you all for your attention.